Hello everyone. So in this presentation we will continue with the structure of DNA as proposed by Watson and Crick and the basis, the studies upon which their structure elucidation was brought about. Now this shows the double-stranded DNA, and here you can see the DNA. It is a right-handed double helix. In the sense they turn in a right-handed manner and around a central axis. You can see over here it is a double helix because it is formed of two polynucleotide chains running in a helical way. Okay. Now it consists of two polydeoxyribonucleotides twisted or turned around each other around a common axis. Okay. The two strands, they are anti-parallel in nature. As you can see over here, this is one strand which here you can see it is 5' and this is 3' in this direction. And the other strand with 5' and 3' in the opposite direction. Okay, that is the 5' end of one chain, it actually faces the 3' end of the other chain. So it is anti-parallel in nature. Okay, so the one strand, it runs in 5', 3' direction, while the other in the opposite way, right? So now the other features, if you see, you can see that the width of the diameter, okay, the width or the diameter of the double helix, it is 20 angstrom in diameter, or you could say it is 2 nanometer. Okay, and each turn, so the turn, if you see, it's a single turn, it extends from here till here. Okay, so this is one turn. Okay, so it extends from, for example, it is marked like this, isn't it? So it will turn, it will go this way, right? And then the other, it goes around the central axis and comes in front and then ends over here. So this is one turn. Okay, and this one complete turn, it is known as the pitch of the helix. Okay, and the pitch of the helix, it is 34 angstrom or 3.4 nanometer. And each turn has 10 base pairs of nucleotides. Okay, 10 nucleotide base pairs, and each adjacent pair, it is placed 3.4 angstrom apart. Okay, so that is how it is. It is total of 34 angstrom. A single turn is 34 angstrom. It is known as a pitch. Okay, and there are about 10 base pairs in single turn. So two adjacent base pairs are 3.4 angstrom apart. Okay, now each strand has a hydrophilic deoxyribose phosphate backbone. As we have, you can see over here, phosphate sugar, this forms the backbone. Okay, and this is hydrophilic in nature. Phosphodiester bonds, they are hydrophilic in nature. Hope you remember, it do have O minus, O negative charges, isn't it? Two O negative charges which are free. So that actually gives a what you call hydrophilic nature to the phosphodiester bonds, and this hydrophilic phosphate sugar backbone, it is in contact with the external watery medium, and they are set on the outside. They are periphery of the molecule. They are found on the periphery of the molecule. They are in contact with the aqueous watery medium. Okay, I mean external medium. Now, what about the nitrogenous base pairs? The base pairs, they are hydrophobic in nature and they are stacked inside. They are inwardly directed and they are stacked inside. Okay, so this actually provides them a stable configuration. Now, as already mentioned, the two chains, the polynucleotide chains, they are not identical. Instead, we call them as complementary with each other. So here you can see they are not identical, isn't it? So wherever you have G on one of the nucleotide chain, you will find C on the other. That is guanine having cytosine on the other. Similarly, you can see over here, isn't it? Cytosine with guanine. Wherever we can find thymine in one of the strands, adenine will be on the other. So there is a complementarity. Okay, so it is due to the base pairing. So the two polynucleotide chains, they are complementary to each other. They are anti-parallel, they are complementary to each other. Okay, now these two strands, the polynucleotide chains, they are held together by way of hydrogen bonds between the complementary base pairs. You can see guanine base pairing with cytosine through 3 hydrogen bonds, while the thymine base pairs with adenine by way of 2 hydrogen bonds. And always there is what you call a pyrimidine purine bonding, and specifically adenine with thymine and cytosine with guanine. Okay, so since there are 3 hydrogen bonds between guanine and cytosine, this is far more stronger than the thymine AT double bond. Okay, A double bond T, that bond is just only is two bonds are there, isn't it? So what happens is GC bond is more stronger than the AT bond, and hence whichever part of the DNA has more amount of GC bond, it will be very difficult to break the hydrogen bonds or denature them. Okay, get it separated. Okay, the GC is stronger by about 50% than the AT bonds. Okay, now as already mentioned, the AT and TA, GC, CG, all these are very specific with respect to double-stranded DNA. Now, these complementary base pairing, it was done or it was taken into consideration while detecting that this particular double helical structure. These studies were based on the studies conducted by Erwin Chargaff and his colleagues. Okay, and that was popularly known as Chargaff's rule, which we'll be looking in one of the coming slides. Okay, now here you can see this base pair, the sequence of base pairs. It is what actually the genetic information is all about. Okay, so genetic information is stored in this sequence of bases, right? So this sequence of bases, it represents genetic information, and the genetic information is well preserved and stored and protected within the sugar phosphate backbone. Okay, so that is about the double helix, which we have already mentioned, and this is the Chargaff's rule. Okay, Erwin Chargaff and his colleagues, they studied the amount of each of the bases, the base composition, as we can say, of different kinds of what you call different species of animals and plants and even other smaller prokaryotes. Okay, and they found that in every DNA they had almost equal number of adenine and thymine residues and almost equal number of guanine and cytosine residues, and this is what is referred as the Chargaff rule. So I repeat, that is while studying the number of what you call the different bases in the DNA collected from different species, from eukaryotes to prokaryotes, Chargaff and his colleagues found that the number of adenine and thymine residues were almost equal, and similarly they could also find equal numbers of guanine and cytosine residues, and this is popularly known as Chargaff's rule, or it is otherwise known as the molar equivalence between purines and pyrimidines in DNA structure. Okay, so that rule. Okay, so here this particular table, it shows the molar equivalence or the base equivalence in DNA, and here you can see how much mole percent of each of these bases are present in the sample collected from different species, and they also had found the ratio, the base ratio, and it is almost equal to 1. You can see over here it is almost equal to 1, and another important feature is that it is specific. Okay, this particular ratio, it is specific to each species. That is one important feature which we'll see. So Chargaff's rule states that base composition of DNA, it generally varies from one species to another. Okay, you can see over here in humans it is 1.52, while in sheep it is 1.36, like right. But it is all like around 1 it is. But it is specific. To one species it is specific. It varies from one species to another. And DNA specimens isolated from different tissues of the same species have the same base composition. As per Chargaff, they states that from whichever part we have collected the sample for extracting or isolating the DNA, the base composition will be the same, irrespective of the tissues from where the DNA specimens were collected. Okay. Now, the base composition of DNA in a given species does not change with an organism's age, nutritional state, or changing environment. So it is highly specific. The base composition of DNA in a particular species, it is specific. So what does it say? It may change from one to the other species, but it may be same in every member of a particular species. Okay. In all the cellular DNAs, regardless of the species, the number of adenine residues is equal to the number of thymine residues, and the number of guanosine residues, it is equal to the number of cytidine residues. And from these relationships, it follows that the number of purine residues will be equal to the number of pyrimidine residues. Okay, so that is how they have come up. So A plus G, that means the total amount of purine residues present in the sample will be equal to the total amount of pyrimidine residues in the sample. Okay, so Before going into this one, actually, when Watson and Crick came up with the structure of double-stranded DNA, the what you call double helical structure of DNA, as it is popularly known as double helical structure of double-stranded DNA. The structure was based on the studies or the results obtained through the Chargaff study, as well as the studies conducted by Rosalind Franklin and Morris Wilkins. Uh, Rosalind Franklin and Morris Wilkins, they conducted X-ray diffraction studies, and the results obtained were useful for like determining the double helical structure. While the results obtained through Erwin Chargaff's study, it was useful for developing the what you call base composition, as well as what you call the base pairing, the specific base pairing, complementary base pairing, which was used for developing the double helical structure of DNA. Okay. Now, what are the different factors that stabilize the DNA double helix? One is, as already mentioned, hydrophobic interactions. Hydrophobic interactions between the nitrogenous bases, the stacking interaction again between the stacked bases, that is between the base pairs. Okay. Now, hydrogen bonding, it is between the bases, and also the hydrophilic interactions of the what you call the phosphodiester bond with the surrounding watery medium. Another interaction which stabilizes the DNA double helix, it is a charge-charge interactions. That is electrostatic repulsions of negatively charged phosphate groups. It is minimized by the interaction with cations which are present in the surrounding medium. That is in the what you call the cellular fluid, there will be cations like magnesium or sodium or calcium. So such kind of cations will actually interact with the phosphate groups, and hence bring down or minimizes the negative charge. Okay, so this will also help in stabilizing the DNA double helix. I hope it is clear. So we in this presentation and the previous one, we discussed about the structure of DNA, the Watson and Crick model of DNA, that is the double helical model of DNA, which describes the structure of DNA and how the sugar phosphate is arranged, and how the nucleotide base pairs are arranged, and what is the pitch, what is the diameter, and what are the distances between the adjacent base pairs, etc. Okay, and here we have concluded with the factors that stabilizes the DNA double helix, and why the DNA double helix or double-stranded DNA is more stable. Okay, fine. Thank you.